Welcome, everyone. You are going to hear about investing in Canada's nanomedicine ecosystem. I'm Marie Diorio. I'm the president of NanoCanada, and I work at the University of Ottawa. I am delighted to have the Nanomedicine Innovation Network, NMIN, as a partner in this event, and also to have Western Economic Diversification support the event. And of course, I would like to thank in advance our speakers and panelists who are made themselves available this afternoon to for this very interesting program that you're just about to witness. And a special shout out to those participants who are joining us from different countries around the world. So thank you all. And now it's I'm very happy to introduce our first keynote speaker of the day, Dr. John Lewis. Dr. Lewis holds a Frank and Carla Sajonki Chair in Prostate Cancer Research at the University of Alberta, and he's a professor in the Department of Oncology, where he chairs the Alberta Prostate Cancer Research Initiative. He's also the CEO of Entos Pharmaceuticals, an Edmonton-based biotechnology company developing genetic medicines using the Fusogenics drug delivery system. Entos is developing DNA-based vaccine and therapeutic countermeasures for COVID-19 using the Fusogenics platform technology. It's my pleasure to invite Dr. Lewis to share with us his talk, Ushering in a New Era of Genetic Medicines. Dr. Lewis.
Thank you, Marie. That was a wonderful introduction. Really appreciate the invitation to participate in what looks to be just a fantastic program. So I'd love to spend a little bit of time talking today. I've got a really bold title because I think I'm really excited and enthusiastic about the future. It's been a really tough year for many of us, but one thing that's become clear from the first effective COVID-19 vaccines is that we're in a completely new era of being able to very rapidly develop safe and effective medicines for a whole new range of diseases. And I'd like to talk to you about how Entos and my academic lab have been working on technologies to contribute to this area. So quickly, I know a big theme of NMIN and NanoCanada is really the bridge between the academic research, discovery-based science, and commercialization and spin-off companies. So I just wanted to give you just a sort of a high-level overview of my program and how I approach it. So at the University of Alberta, through the prostate cancer chair, we run a province-wide translational research program called the Alberta Prostate Cancer Research Initiative. And our goal with this was really to bring together, the academic side of things, so principal investigators and researchers in the field of cancer and prostate cancer and other things, and really join them with the physicians working in cancer, the clinical trials folks, and everybody else who's involved in that spectrum and continuum between discovery and application. And so through that, we've got a lot of people together in Alberta, and through an international network of collaborators, we've identified several very important things. One of those was to create a biobank and a registry. So we have a registry. Every new diagnosis of prostate cancer in Alberta gets enrolled in our registry. We collect biospecimens. We're networked with 14 countries through Movember to be able to collect and follow these patients over time and collect their outcomes information. And then through the technologies and endeavors of the academic scientists in the group, we've then identified key technologies and spun those out as companies. Nanomedicines is a diagnostics company. Entos, I'll be talking a lot about today, is a genetic medicines company. Oncogenics is a cancer company, and Aegis Life is an infectious disease company. And so what's nice is the trainees in these programs and the physicians and PIs working on different parts of the program have really been a functional part of taking this technology to the next step. So Entos Pharmaceuticals, as Marie said, is an Edmonton-based biotechnology company. It's founded by myself, our CSO, Arun Raturi, and Roy Duncan, another Canadian scientist who really discovered the underlying technology. Also, Colin Corus is a key part of our team, and we have sort of this great, science-based team based in Edmonton. A lot of advanced degrees and really focused on the science of genetic medicine. We're in the Merck Invention Accelerator facility in Edmonton, Alberta, but we also have a presence in the U.S. in San Diego, part of the J&J labs in San Diego, and we're closely affiliated with the Alberta Cell Therapy Manufacturing Facility in Edmonton, where we can, we're scaling up manufacturing GMP quality material. And so I thought I'd start with a high-level view. I mean, at Entos, we're thinking big. So we imagine this world where we're free of all these genetic diseases, and I think if you look at the underpinnings of most diseases, there's a genetic component. So I think we've really seen and really propelled ahead with the pandemic this new reality coming into focus. One that'll be ushered in with the advent of safe and effective redosable nucleic acid delivery technology. And there's very many people in the NanoCanada and NMIN groups that are keenly interested in and working on technologies that can create this future. And I would say, really this year, and it's really been building up for the past 2 to 3 decades, the age of genetic medicine is here. And it's really been the innovation and delivery platforms that have been the key enablers of broad application of genetic medicines. And broadly, this is very broadly as an introduction, we sort of have 2 ways of delivering genetic material in the body. One are viral based, so viruses have evolved for millions of years to be very good at delivering their genetic material. So we've been able to hijack this to deliver therapeutic payloads. And we have approved viral gene therapies. This has really spawned a huge industry in viral-based gene therapies. And really, I would say they work great, but their Achilles heel, the real challenge for viral vectors are redosing because they're immunogenic. On the other side of things, we have the scalable non-viral technologies. I would say at the forefront of this are lipid nanoparticles. The technology developed in Canada, really bringing, really the underpinning the huge successes by Pfizer, Moderna, BioNTech on vaccines this year. And we've had through companies like Alnylam approved systemic therapies based on LNP technology, and now with vaccines. And I would say LNPs are great as far as their ability to scale and their ability to deliver, but they're not perfect. So I think there's some iteration that can be done, and sort of that's the field we're working in. And I would say on the whole, these genetic approaches have now made the world's most highly effective vaccines. And I would say this is really just the beginning of a huge wave. And so I think, many different delivery platforms we brought to bear depending on the application. And at Entos, we thought, what if we could take the best part of the viral approach and combine it with sort of the scalability, redosability of the lipid nanoparticle approach. And if you look at lipid nanoparticles, these have been iterated and developed over many years based on efforts, mainly in Canada. But the idea here is that we have several lipids in a mixture that both encapsulate, so neutralize the charge of nucleic acids, the negative charge of nucleic acids, to be able to compact them and encapsulate them, but then providing stability. Providing enhanced biodistribution, where depending on the route of administration in the body, but most importantly, then the delivery. So ionizable. So originally, cationic or positively charged lipids were used to basically when they would interact with cells, get taken up in endosomes, would be able to escape that through positive charge. The most recent development of ionizable lipids that remain neutral in the bloodstream and then become positively charged and change their conformation in the endosome to facilitate escape is the new technology. And so this is really the underpinning that now of the current vaccine technologies and other technologies being developed for a variety of different diseases. And I would say that spectacular approach that is really limited by this positive charge aspect. So the sort of the Achilles heel, or the challenge with the positive charge, that it also produces some tolerability issues when given in high concentrations. And for systemic dosing, this has really been a challenge for viral and non-viral based therapies, is that they accumulate in the liver and you get these dose limiting toxicities. So at Entos, we thought the ideal LNP platform might be one that uses an alternative mechanism for delivery. And this really stemmed from this discovery by Roy Duncan at Dalhousie University a number of years ago of a really interesting class of orthoreovirus called a fusogenic orthoreovirus. Now, this doesn't affect humans. It affects things like birds and alligators and baboons. But this virus does a very interesting thing when it enters cells. So it doesn't use a fusion protein to enter cells, but once it infects them, it rapidly causes all of the cells around them to fuse together. And I got this little movie here to show it. And what Roy realized at the time that he had discovered sort of something really novel, a novel fusion protein, but we didn't realize is how unique it was. So I'm showing here, so many viruses have fusion proteins that they utilize to facilitate cell entry. So this is the flu HA fusion protein. It's anchored into the membrane. It's got multi-subunits. It's quite large, but it's elegant in the way it can bring two membranes together and fuse. What Roy realized is these orthoreoviruses expressed what he called a fusion-associated small transmembrane protein, a completely new kind of fusion protein that's extremely tiny. So 2 orders of magnitude smaller than HA, single transmembrane domain, tiny ectodomain, that would help with its immunogenicity. But most importantly, it's sufficient to induce membrane-to-membrane fusion. And if you formulate it in a liposome or lipid-based nanoparticle, you can facilitate direct fusion of the lipid nanoparticle with the plasma membrane of target cells. And Roy spent the better part of 2 decades discovering and characterizing the fusion-associated small transmembrane proteins from a wide variety of different orthoreoviruses. And then in collaboration with him, we've basically taken the best, most functional parts of each to combine them to create fully hybrid manufacturable fusion proteins. And I'll just show you a snippet of the kinds of things we've been doing. So this is the fusion protein here. It's a single transmembrane domain with this ectodomain that's anchored through a myristylated post-translational modification. So this tiny, this is the fusion action domain. And then this transmembrane domain, the C-terminus is mostly required for viral pathogenesis. So maybe dispensable for fusion. And so we've sort of mixed and matched the different components, the different domains from these proteins from the reptilian and the baboon version of this fusion protein to see where the activity lay and see if we could increase the activity. What we found generally is that we either abolished the activity completely or it was pretty much the same as the parent molecule. But in one case, when we took the ectodomain of P14 and fused it to the endodomain of P15, we actually got a molecule that was much more highly active to fuse cells together than either of the parents. And so the P14 endo P15 fast protein is currently the one we use in the platform. And so we've dubbed this platform a proteolipid vehicle because it's essentially lipids mixed with this fusion protein to deliver nucleic acids. And so we've been working for many years on how to make these in a reproducible, stable environment. We've developed a single-step microfluidic manufacturing process, which incorporates lipids, fusion protein, and cargo all at the same time. And we've scaled this up now to GMP. And now, because we're using a fusion protein to do delivery, it's given us quite a bit of flexibility in the lipid components. So we've been able to optimize formulations to encapsulate a wide variety of different nucleic acid cargoes, including small things like RNA, siRNA, messenger RNA, plasma DNA, even big things like proteins and for gene editing, et cetera. And so as I'll show you, surprisingly, even though this is a viral protein, it's non-immunogenic, so we can repeat dose. And again, we have the scalable, like LNP is a scalable and inexpensive manufacturing. So the one thing we got really excited about is that we were able to effectively deliver larger payloads like plasmid DNA. I'm just showing you here several examples of being able to take sort of a neutral lipid formulation, incorporating fusion protein and how that changes its ability to transfect a wide variety of primary human cells, so human fibroblasts, liver cells, brain cells, endothelial cells, so blood vessel cells. And once you incorporate these fusion proteins, we get really, really high transfection rates. So the ability to transfect a wide variety of human primary cells. We can also encapsulate RNA as well. So when we encapsulate RNA, for instance, in primary cell lines like retinal epithelium, for instance, and we can get a dramatic increase in the ability to transfect with RNA when we incorporate these hybrid fusion proteins. And so thinking about the kinds of platforms, the kinds of therapies we can develop, the route of administration is really important. So what's nice is that because we've been able to remove some of the ionizable lipid, we've been able to remove things like cholesterol. We actually get pretty good biodistribution after systemic or intravenous injection, which is nice. We can hit a variety of different tissues. Upon intramuscular dosing, such as we're doing with the vaccine, we get good, durable, strong, but localized expression that doesn't become systemic. And what's really interesting in an area of excitement and development for us right now is actually when we deliver it orally, we get really actually very good expression throughout the GI tract and beyond. So this really opens the door in the future for orally delivered genetic medicines. And so, I mentioned the viral fusion protein. We expected it potentially to be quite immunogenic. Producing an immune response against the platform instead of the cargo, which would, for viral-based platforms, really prevents you dosing more than once. And so in this experiment, we encapsulated a messenger RNA expressing luciferase, so it produces light, injected it intramuscularly every month for 5 months. And then interestingly enough, we actually get an increasing expression over time. This is probably due to residual material left at the site over time, but definitely there's no production of antibodies that neutralize the response. And we did this both intravenously and intramuscularly. And when we look for, in these animals after 6 months, levels of anti-fusion protein antibody levels, we find basically nothing, so barely above the level of detection, even with an IV repeat dose over 6 months. And what was interesting, though, is if we looked at the production of antibodies against the cargo protein, in this case luciferase, we actually saw very, very high titers. So this got us excited about the application, potentially this platform for a vaccine. I'll just quickly say we have fantastic collaborations on the manufacturing side. We're working closely with Precision Nanosystems in Vancouver, and you'll hear from Lloyd Jeffs later on today. We've got a great scalable microfluidic chip-based platform that allows us to prototype and pilot formulations at the benchtop, scale these up to in vivo studies and large animal studies, and then go straight to GMP manufacturing, which we've now done with the vaccine, which is very exciting. And then we're working closely with the Alberta Cell Therapy Manufacturing Facility at the University of Alberta, where we've just made the first GMP batch of COVID-19 vaccine. So through these partners, we've been able to bring a technology all the way from discovery to the clinic. So Entos really approaches fusogenics as a platform technology. Obviously being an oncology researcher, I'm passionate about cancer. So we have spun out a company called Oncosenics to go after cancer, and I'll show you just a little bit of information about Oncosenics. We're working with age-related diseases, which is really exciting. I'll show you a bit of data from that as well, and now vaccines. But we're working with over 24 of the world's largest pharmaceutical and biotechnology companies on sort of their next generation therapies, really moving the world, moving the needle from small molecules and even antibodies to genetic medicines. And so we're really excited to work with a number of great companies on these kinds of technologies. Let me just show you a couple of really exciting things before we get to the vaccine. Ocean Biotechnologies is focused on senolytic gene therapy. This is the idea that as we age, we accumulate these cells that have damage. To react to this, the cell puts itself into a program called senescence. But senescent cells don't die. They just accumulate and they secrete factors that actually facilitate aging and a lot of the age-related diseases. So I don't have a ton of time to go into it. But if the idea here, it's been shown by a group at the Mayo Clinic that if you are able to ablate senescent cells in a mouse, for instance, you can extend lifespan by over 20%. And so we developed a gene therapy, basically a programmable gene therapy that can selectively ablate senescent cells now through an IV infused fusogenics formulation. And we actually showed when we, in combination, took 2 senescence pathways, so a P53 based pathway and a P16 based pathway, and ablated those cells, we were able to extend median lifespan in C57 Black 6 mice by over 20%, basically replicating that Mayo study, which is super exciting. So we're taking these formulations to the clinic for a variety of different age-related diseases. Oncogenics is of course, my main passion is cancer. I mean, we make the bold statement, this is the first viable gene therapy for cancer. Again, this is a programmable suicide gene-based therapy that goes after key oncogenic signaling pathways. And recently we completed a study in colon cancer. This is a syngeneic model, so this has an intact immune system. And when we treated either as a single agent in this model or combined it with checkpoint inhibitor. So the single agent outperformed checkpoint inhibitor, but when we combined the two, we got complete cures in all of the animals. So really excited about this. We're working closely with a group at the Cross Cancer Institute in Edmonton, Alberta. This is supposed to be in clinical trials already, but COVID hit and put our plans back a bit, but we're hoping to get this into a phase 1 trial this year. Okay, so I'll spend the last few minutes talking about our COVID-19 vaccine program. So we've got go back to March 2020. We've got this platform we've really optimized for the delivery of gene therapies, DNA, and looking at, so the programs are being developed in RNA. We recognize that one of the limitations would be storage and distribution. So we decided to basically use similar biology, but then go after a DNA vaccine. And what we were hoping, and this is through our design considerations, we'd be able to attain, really good stability at fridge temperatures for over a year. Because DNA expresses the antigen in slightly different kinetics, we expect it to be effective potentially after a single dose. Like RNA vaccines, DNA vaccines produce a durable T cell response, which is emerging as a key mediator of durable response. And of course, with DNA like RNA, you can incorporate multiple epitopes. We have a candidate that's a pan-coronavirus candidate. And then we have this, scalable manufacturing, which is required to produce hundreds of millions of doses, for a raging pandemic. So we looked at about 24 different candidates and narrowed them down to 2 of them. I'll just quickly go over it. So the first one, very, very similar to the approved, effective, safe vaccines that are out there, except DNA. So it encodes the full-length spike protein. It's encoded in a really sort of cool nanoplasmid technology with our Partridge Nature technology. And because it's a genetic vaccine, we've been able to incorporate genetic adjuvants, which are really neat. So a double-stranded RNA CpG adjuvant and a double-stranded RIG-I agonist adjuvant. And so, so we know that safe and effective vaccines can be built on full-length spike protein, and we've got some great data for that. VAX-002 is our next generation, sort of second generation vaccine. We use machine learning to identify 34 optimized epitopes, and not just from spike protein, but also from the envelope and membrane proteins, which are far more conserved than the spike protein and should prevent, escape from protection by variants. And so again, this is we have some really nice preclinical data for VAX-002. I'll quickly go through this. So when we immunize mice with either candidate, we basically so if you think about the DNA vaccine world, Inovio and others are developing DNA vaccines, but because they're using either electric shock or a pressure-based device to get it in, their doses are very high. So 1 milligram per dose times 3 doses, 2 milligrams per dose times 3 doses. And we were able to get durable protection or seroconversion at doses as low as 25 micrograms with both candidates. This is the antibody response. When we looked at the neutralizing ability of these antibodies, do they prevent SARS-CoV-2 infection? We're able to get really high neutralizing titers, as high or higher than what we see in convalescent patients. We've also looked at the ability to generate neutralizing titers against some of the variants out there. And so we're actually better than convalescent patient sera in neutralizing ability of the variants, and we're continuing to look at variants as they emerge. This is probably the most exciting data for me when we vaccinated African green monkeys and then looked at the neutralizing titers over time, whether giving a single dose or 2 doses spaced 28 days apart. We actually saw a steady increase in neutralizing titers going over this expected protected level after 28 days and continuing to go up. So this data is still coming in. But what's really neat is that both the single dose and the 2-dose regimens basically gave the same neutralizing titer. So we think a single dose would be sufficient. Not to miss T cells. So VAX-001 produces a very strong and potent spike-specific T cell response. That's basically a CD8 killer T cell response, which is the kind of response that we were looking for. When we actually generated a functional T cell killing assay, we're able to show that T cells from immunized mice with VAX-001, in a dose-dependent manner, we're able to kill target cells that express the spike protein. So we know we're getting, production of these CD8 T cells that are able to kill target cells. Very exciting. And we've done this now in a challenge model, working with Vito in Saskatchewan on the hamster challenge model, in a single dose and 2-dose regimen. We're seeing, so hamsters, when they get SARS-CoV-2, they lose a lot of weight. So usually up to 20% of their weight gets lost in 5 to 6 days. If you can reduce that weight loss, that it correlates well with viral titers. We're getting substantial, significant improvement in the weight characteristics in hamsters. This is early data that's still reading out, but really it seems like the vaccines protect well against infection in this model. And so we're just on the cusp of starting a phase 1, phase 2 clinical trial at the Canadian Centre for Vaccinology in Halifax. We'll enroll in the phase 1 portion 72 participants, and the phase 2 portion 500 participants, and we'll be looking at things like safety and efficacy in both young healthy adults and elderly, looking at multiple dose levels, as well as the immunogenicity of both single doses and 2 doses. So it was a lot of material, and I tried to stay in the time. And I think, it's been a super crazy year. The team has worked really, really hard on this vaccine. What we're seeing, both through the efforts of many companies in this space and our own, is that the future is really bright for genetic medicine. With respect to the pandemic, I think what we're finding, initially we thought it would be a race to a vaccine and then we'd be out of it. It's looking more like a marathon. But that's fine, because there's been enough investment and innovation around the world that we have the tools to be able to win this race and this marathon. But we're looking, the task before us is staggering. We need at least, not even including variants, we need to get up to 16 billion doses in arms worldwide. We see these novel variants that are now escaping the first generation vaccines. So it looks like this virus is likely to become endemic, and we'll require yearly boosters. And it's really incumbent upon Canadians and Canada to invest in both the innovation and the manufacturing to be able to continue to respond to this. I think what's really exciting about what we've shown in these different projects is we can now essentially write biological software to cure or prevent disease. For me, that's really exciting, and Antos and the other teams we're working with are really looking forward to seeing how far we can take this, because I see a world where no disease is out of our reach. So I'll end there. Many, many people to thank, a great team, lots of partners, and appreciate NMIN and NanoCanada for the opportunity to talk. Thank you.
Thank you very much, Dr. Lewis. I think we have time for 1 or 2 questions. And I think if you use the Q&A button at the bottom, Marshall can unmute you if you have a question.
Yeah, feel free to raise your hand when we do have a question. Karat, I'm going to unmute you. If you unmute yourself, you should be able to speak. Go ahead.
Hi, Dr. Lewis. Very well talk, and I really like the fusion protein, and that is one of my growing passion. I'm working with the LNPs right now. So my question is, in one of the slides you mentioned you guys are working with the stem cell reprogramming. I'm interested in that. I want to know what kind of reprogramming you guys are doing.
Sure. No, it's a great question. So as I mentioned, we're really passionate about age-related disease, and stem cell reprogramming has emerged as one of the key, so we can, senolytics can potentially take away the damage, but unless we repopulate those stem cells and rejuvenate tissues, we're going to reach our limits. So we're working with some of the world-leading groups that emerge out of Stanford, for instance, looking at mRNA-based multi-target rejuvenation of stem cells, basically reprogramming them, not completely reprogramming them to a pluripotent state, but transiently reprogramming them to a younger state to enable tissue regeneration.
Okay, very well. Thank you so much.
Thank you very much for your question. I'm afraid we don't have more time. So Dr. Lewis, thank you so much for kicking off the event with such an amazing keynote talk.
Great. Thanks, Marie. Pleasure.
Thank you. Now, I'm pleased to introduce the moderator of our first panel discussion, Dr. Nuzhat Tamzaman. Dr. Tamzaman is a pharmaceutical scientist and an entrepreneur. Together with her husband, Dr. Y.K. Tam, she founded Sinoveda Canada in 2006. She presently leads the business development and the marketing and distribution of Sinoveda natural health products in Canada, the US, Asia, and the United Arab Emirates. She assists global consultants to register and market Sinoveda products in their respective jurisdiction. It's my pleasure to invite Dr. Tam Zaman to introduce the speakers on our panel on building Canada's advanced manufacturing capacity in nanomedicine. Nuzat, the floor is yours.
Thank you, Dr. Delorio. I'm pleased to introduce our three panelists, Drs. Lloyd Jeffs, Lakshmi Krishnan, and Trina Racine. Dr. Jeffs is a Director of Clinical Manufacturing Solutions at Precision Nanosystems, Inc. He brings 20 years of experience in the biotechnology industry, working on lipid-based drug delivery systems, and has previously worked for Northern Lipids, Inc. and Arbutus Biopharma Corporation. Dr. Krishnan is the Acting Vice President, Life Sciences of the National Research Council, as a globally recognized researcher and expert in the field of infectious diseases and cancer. She is committed to the development of novel vaccines and therapeutics and innovative solutions for the improvement of human health. Dr. Racine is the Associate Director, Vaccine Development at the Vaccine and Infectious Disease Organization, or VIDO. She has worked on the development of vaccines and therapeutics for various emerging and re-emerging infectious diseases, including Ebola, Zika, and MERS. Prior to joining VIDO, Dr. Racine was a scientific and regulatory affairs consultant for Gene One Life Science, Inc., a South Korean-based pharmaceutical company. So welcome, Drs. Jeffs, Krishnan, and Racine. So let's go to my first question, and that is for Dr. Jeffs from PNI. PNI has received funding from the government recently for both developing and manufacturing vaccines. My question is, will this funding be enough for PNI to address the present pandemic? If not, what are the gaps that need to be addressed? Or simply put, can we have a made in Canada vaccine? A question probably that is in everybody's mind today. So Dr. Jeffs?
Anyway, thank you very much for the introduction. I think that's a dream of mine and a dream of everybody on this call to have a made in Canada solution, COVID-19 vaccine. Having said that, I think importantly, the great work that's been done over many years with lipid nanoparticles and their development originated in Canada. So we have basically started this progress, and through the great work of hundreds of researchers from the University of British Columbia, where I studied and worked originally. I originally started working in Dr. Tom Madden's lab and Dr. Peter Cullis' lab while working at Northern Lipids. And then you could see how those innovations have continued and been built upon the years to the point now where we have approved lipid nanoparticle-based vaccines. What I would really like to see is those vaccines to be actually manufactured in Canada. So currently they are manufactured and for clinical and for commercialization outside of the country. So I think that's the missing piece that I'm focused on at Precision Nanosystems, to build that capacity for commercial manufacture of genetic nanomedicines, whether they're vaccines or therapeutics, and supporting our partners and supporting our clients to enable that, to provide the tools and the services to produce material for clinical trials and ultimately for commercial production and distribution. With respect to the amount of funding, it's very welcomed. The Canadian government contributed $25 million to the building of what P&I is calling it, the Biomanufacturing Center for RNA medicines. It's a $50 million project in total, and we will have that completed in early 2023. So that will mean that the innovations that are taking place in Canada, GMP manufacturing can be supported at our facility, and we'll have a roadmap to support commercial manufacture. And because of the technology we're using, it's small footprint. I think there's also potential for distributed manufacturing throughout centers in other parts of Canada and other parts of the world. Because I think what we're seeing is, even with the vaccine approved, distribution, the supply chain part of the vaccine and therapeutics, especially for these emergencies, emergency events like pandemics, is a challenge, right? It's a challenge to import, to secure supply. I think the Canadian government's done a fantastic job actually of doing that in light of the fact that we do not have domestic vaccine manufacturing. So I think we've done as best we can, but for the future, it'd be great to have that in place.
Thank you. Thank you, Dr. Jeffs. So my next question is for Dr. Racine from VIDO. VIDO is a global leader in infectious diseases and have all the in-house capabilities to develop vaccines. Recently, you received funding from your local government, excuse me. Previously, you have worked with Zika, Ebola, and MERS viruses, and now working on COVID-19. VIDO was the first one to isolate the virus and test it in an animal model. So where are you at now, and how can you be ready before another potential pandemic? Dr. Racine?
Thank you very much. And I just wanted to also say thank you to the organizers for inviting me today. And so you're right, we were the first organization in Canada to isolate the virus in collaboration with the Public Health Agency of Canada and a group in Ontario. And we were also the first to develop the animal model, ferret to ferret model, and now a hamster model, which enabled us to, start developing our own vaccine in-house, but also to work with outside groups such as Entos to help develop their products, whether it be vaccines or treatments. And so for us internally, our vaccine, one of our vaccines is currently in a phase 1/2 clinical trial being conducted in Halifax at the Canadian Centre for Vaccinology. So that's very exciting news for us, but we're not stopping there. We've got a second vaccine that we're also going to hopefully bring into the clinic shortly. And then of course, move on to higher level clinical trials. And so part of, what we have done wasn't actually hampered by the fact that we didn't have in-house manufacturing capabilities at the start of this pandemic. We could have potentially been in the clinic a little bit sooner had we had the ability to do some of that process development, prior to the pandemic. And also, while thankfully we have been able to partner with a number of great Canadian organizations to do some manufacturing, having that ability in-house would have been quite beneficial to us. And so thankfully, the federal and provincial governments have provided funding for us to build a manufacturing facility here at VIDO, and the construction for that is underway. The demolition part of it is now complete because we are building the facility within our current existing Intervac building, which is our level 3 containment facility. And right now we're on track to have construction completed by October or November of this year, at which point we will start commissioning the facility and bringing in all those, large pieces of equipment that are required to produce vaccines. And so, and with the goal of being, operational by 2022. And a lot of that, the vaccine facility is going to be not just a one platform facility. We're not a commercial scale facility. We're going to be a pilot scale manufacturing facility where we'll be able to work with outside organizations who have different products, whether it be a live attenuated vaccine, an inactivated vaccine, a subunit vaccine, a viral vectored vaccine, whether it be for veterinary use or human use. And so I think having a facility that has this capability of being able to work with multiple platforms will enable us to be even more prepared for the next outbreak, because, you know, an RNA vaccine this time, has gone into the clinic and been authorized really quickly, which is fantastic, but maybe an RNA vaccine may not work as well the next time. Or if it's a veterinary outbreak, you know, there's some African swine fever comes to Canada, and, you know, now we need something for that, you know, group. Well, then we have the capacity to be able to be producing vaccines for various reasons. And so I think that's a really great thing that we have to our advantage.
Okay, thank you. Just a follow-up question I wanted to ask is, let's say the government has invested into this program, but if they invested the same amount of money that US and UK did, do you think that we would be able to have a vaccine on time?
Well, I think, you know, so the amount of money is definitely important, but it's the timing of that money is even more important. You know, throwing $1 billion at us in March of last year maybe might have been able to make things go a little bit faster, but not a whole lot, because we still needed to work with outside groups to be able to produce anything, because we didn't have the capacity internally. So had that money come into play 5 years ago and we had the facility already up and running and we already had all the capacity here, then yes, absolutely, things would have been much different.
Okay, thank you. Okay, now my next question is for Dr. Krishnan. What is the overall role of NRC and its collaboration with various Canadian technology and innovation companies, and how leveraging this Canadian expertise could lead us to have a vaccine made in Canada in the very near future? Dr. Krishnan?
Thank you, Nuzit, for the question. And I would also like to thank Dr. Marie Diorio for the invitation to actually speak here today. So NRC has two aspects to it. First of all, we are the largest science-based intramural research arm of the Government of Canada. So we have 14 research institutes that bring together expertise in various areas, and we have a very strong presence in human health. So in fact, in human health, we have the largest R&D team that are solely focused on the development of biologic medicines. So this is 25 years of experience in being able to de-risk and accelerate biological medicines to take it into the next stage of first in human testing, and then even beyond that with our partners. So in fact, we have just since 2012 moved about 11 different biological medicines into clinical testings with our partners. So that's what we do well, work with partners and collaborate and move that to the entire value chain, from early preclinical evaluation all the way to process development, optimization of the manufacturing process, and then technology transfer. So in the event of this pandemic, what we were able to do very quickly is to mobilize our team to help a number of Canadian partners advance their COVID-19 candidates. So for example, we worked with VIDO to help them do the process development of their vaccine candidate to then tech transfer for their phase 1 material production. We are working with companies like VBI, again, to do the same thing, and they have also entered phase 1 clinical trial with the both the preclinical evaluation data package we produced for them, but also with the bioprocess optimization that we did for them. So these are ongoing activities that we will continue to do. But what is more exciting is that we are actually we're funded to put together the biomanufacturing facility. So we started this work in July 2020. And in fact, our new building is going to be all completed construction by the July of this year. So in fact, in Montreal at our facility, the outer shell and the inside, the building is now fully completed. And we are very rapidly setting up all of the interior equipment, validation and commissioning to have a manufactured full production facility for manufacturing vaccines. And this should be operational to be able to be producing vaccines by the end of this year or early next year, once we have all of the Health Canada certification. So I think this is just remarkable speed with which we were able to accomplish this. And this is the first, I think, the step in terms of being able to start with the manufacturing footprint in Canada. And we're looking to, again, collaborate. We have announced a few collaborations, for example, with Novavax, that we will be doing manufacturing. But I think our future looks brighter, because we are going to have also, to complement that, parallel clinical trial manufacturing facilities. So we'll actually have a continuum from being able to help everything from early preclinical development all the way to clinical manufacturing and to large scale manufacturing. And our approach will be to really diversify products, right? So enable many different companies to work with different types of products, and then move them through the value chain, and really accelerate and de-risk, so that the next time around we have an emergency preparedness that we are able to bring to the table, and we would not be starting from ground zero.
Okay. Thank you. I think that's very important, that what you mentioned, the whole supply chain from the discovery to the clinic. And this requires a very robust supply chain infrastructure. And we know that it didn't quite work this time. So if you can just maybe mention a couple of the key things that we need to do to avoid such a scenario in the future.
Yeah, so I think a couple of key things is, first of all, we need an infrastructure, and this is infrastructure we will need in a way that, nationally we have many nodes and centers that are working in a collaborative mode in order to be able to enable this infrastructure and the missing pieces to complete that value chain. So in that, you have a diversification of portfolio, you have established collaboration, you have established networks that can enable that. A second, I think, important piece is that we need to ensure that we have, talent that we have groomed and we have retained in Canada in order to be able to support all of these initiatives that we are putting together. So I think there again, many of these early investments are going to be critical to have partnerships with the universities, partnerships that are firm in enabling our next generation of high quality personnel to be very effective in translational science, to be effective in working in manufacturing facilities, and to really move the dial from early discovery all the way to retaining that in Canada. So NRC definitely sees ourselves in that role to facilitate that in partnership with many other organizations, particularly the universities and particularly the SME community, to maintain and accelerate a rich pipeline of nanomedicines that can advance forward.
Thank you. I think it's very important to have the HQP that we require, and it is very important not just to generate these HQPs, but to retain them in Canada. A lot of times we see that they don't find the jobs they want, and they move south. And also we see a lot of times that innovations get sold out or bought out by large companies or American or European companies, and that is something that we must ensure that Canadian innovation stays in Canada. I think innovations is the most critical and the most important, and I think the hardest part. But then to scale it and getting the manufacturing and the mass production, all it requires is the right amount of resources at the right time. And I think this is something that we have learned during this pandemic, and I hope that both the government and private sectors will be able to ensure that that happens. So thank you for that. Okay. So some general questions that I would like to throw out there, and any one of you can answer. In a post-COVID era, what would be your strategy to maintain the ecosystem that is being created so that we are ready for a potential attack in the future? Any one of you can answer that.
If you wanted me to take it, I can take a first stab at it. I think it's important. Canada's been forefront of innovation and platform technologies. we talked about the fact that we had, we were the first pioneers even in the lipid nanoparticle technologies. So I think that type of mindset to ensure that we continue to develop innovative platforms and disruptive technologies that can be in some ways agnostic to the final application. So certainly in post-COVID times when there is not an emergency, we can kind of target these type of platform development and innovation towards other types of questions which need to be answered, whether it's rare diseases, whether it is genetic diseases and so on. But then that gives us then a leg up when we face with a situation that we need to now immediately respond. We can then turn and pivot these platform technologies very effectively. So I think we have to continue to push that frontier of innovation to find newer platforms, newer disruptive technologies that can not only make vaccines or biologic medicines efficacious, but also maybe improve the way we deliver them. Do we need centralized manufacturing? Maybe we need point of care manufacturing. So all of these are possibilities that we can explore.
Okay, thank you. Dr. Jess, I think you wanted to say something?
No, it's just agreeing with everything Dr. Krishna was saying. I think what we have to realize is it's going to take a long time to rebuild our pharmaceutical manufacturing infrastructure in Canada. And we made some great steps starting last year, and that has to continue. So it can't be put into mothballs. It can't be put on hold for the next emergency or outbreak or pandemic. We have to build a very vibrant and strong, in this case, nanomedicine industry, because the applications are limitless. From Dr. Lewis's talk, I'm sure Dr. Madden will talk about that in his talk, and Dr. Cullis too. There's so many opportunities here. So I think we should be investing in developing genetic medicines and manufacturing them here. That infrastructure then will be in place so we can pivot and respond very quickly to these emergencies. The platform technologies are validated. They're very powerful. And we do have a base here. We have a lot of really highly trained, innovative scientists and technologists that can make this happen, right? So we have the building blocks. We just have to develop a comprehensive pharmaceutical manufacturing industry here in Canada. Continue to do that.
We just heard from Dr. Lewis, and I heard that he will be working with you in PNI for the manufacturing part of it. So that is a good collaboration, I think.
Definitely, I think PNI, that's our mission, right? Ourselves, we do not develop our own kind of therapeutic products, but we want to support the wider industry, whether it's academic startup companies or established pharma companies, to make that happen. And that is happening here in Canada. Our company's 10 years old. We're a startup at the University of British Columbia, and we've grown exponentially in the last 5 years. So it's a success story, but we have to keep that going. We're Canadian based, and we're working with companies all around the world. So in a way, we can export our innovation, but we do have to keep that infrastructure. We cannot rely on other countries to manufacture our drugs for us, right? Especially with the genetic medicine field. We are pioneers in this field.
And if I may, I agree with everything that you guys are saying, but I think we also need to look beyond just the actual manufacturing facilities, because yes, for sure, that was something that we lacked at the beginning of this pandemic, but we also need to, there's a supply chain issue, right? We need to ensure that Canada has access to glass vials, media, pipettes, right? All of those materials that are required to produce a vaccine and to bottle it afterwards. And so if we don't have access to a good supply chain, then it doesn't matter if we have manufacturing facilities that are up and running or that could be manufacturing a vaccine for us. If we can't get the materials in, then we're still at the same spot. So whether that means we need to encourage some of those companies to come set up shop in Canada, so that we have access in Canada versus having to get supplies from the United States or Europe. That's, I think, to me, that's part of that larger goal of what we need to do in a post-pandemic kind of environment.
We certainly have to scale those supporting companies that help with the supply chain and solutions at the same time as we're scaling our industry. That's
so that there is a sustainability aspect of it, right? So even if you have this manufacturing facility being built right now, then you have to have ways to sustain it so that it is an ongoing industry.
Yeah, and there's a lot of new technologies that support nanomedicine manufacturing and development that may not be nanomedicine companies themselves. I'm thinking about companies like Valnorex with their robotic vial filling systems. You know, that this Canadian technology, it's very innovative, and it allows for very rapid development and manufacturing as well. So we have to look at the supporting technologies that might be coming from life science companies or more engineering companies as well. So that dialogue has to be quite a large discussion involving many stakeholders in the core industry and then supported industries as well.
So you would be working with other companies to provide services once you have your manufacturing facilities set up?
We would look to other companies to supply, you know, say a filling solution, a vial filling solution, supply chain companies to provide the raw materials we need to make these lipid nanoparticles. Precision Nanosystems is looking at making its own drug substance, its own RNA. And that's part of our plan as well. But again, you know, we have to have access to things like bioprocessing bags, the vials, as you mentioned. So yeah, we have to see if that's feasible for that to be done in Canada. And probably the expertise exists. But again, how do we scale that as well?
Okay.
And from a Vito perspective, we are planning to, you know, work with outside organizations that have different, you know, platforms to help them build their process development and whatnot. So that way, potentially, you know, they might want to set up shop in and around our organization or somewhere in Canada so that there's a, you know, easier access to working with them in the future. So hopefully that too will help bring, you know, new innovation to the country.
Yeah,
Absolutely. Yeah.
I mean, you saw example of that even in the early days of the pandemic, for example, when we had a PPE shortage and we saw Canadian companies that were in other sectors that could immediately move to it. So in areas of, you know, advanced materials, we know there are companies doing other types of work. And so part of, you know, some of the IRAP programs that we have launched on innovative challenges have focused in trying to put that challenge out to the, you know, SME community in Canada that may not be in the biopharmaceutical sector right now, but may have other expertise tools that can. So I think that will also become quite important to see how we are resourceful in our approach in ensuring the supply chain within Canada.
Absolutely. I think we know of companies here that make supplements and they just retooled their equipments to make hand sanitizers. You know, just made them stay afloat at the same time, helping Canadians at a time of, you know, this crisis. And same with companies that makes garments and made masks for us. So that's very important to be nimble and quick and to be able to adapt to changes. I think it's very important. A question for Dr. Racine. I know, heard that Vito is, of course, you have all the containment labs, and now you are building a manufacturing facility as well. So my question is, would you work with BNI in the future?
Of course, there's no reason why we wouldn't. We're open to any and all collaborations. You know, the goal is to keep innovation moving forward, you know, and so they have products that, you know, need to be tested in a high containment facility. Well, that's what we're here for. Our, you know, Interback building is one of the largest level 3 agriculture facilities in the world. We can work with animals from mice and ferrets and hamsters to pigs and cows. And we've had bison here, we've had alpaca, you know, so it really there's sky's the limit, right? And so we are open to working with any and everybody. And, you know, we hope that you come to us for that kind of partnership. Absolutely.
So there is a lot of synergies amongst all the Canadian companies that are working towards this great.
Yes, absolutely.
Okay. Well, thank you. So now I think we are almost coming to the end, but one last question from me. If there was one message that each one of you would like to share with the relevant government agency today or any private sectors, what would that be? Dr. Krishnan?
I think from my perspective, I'd say that Canada needs to establish strong partnerships and a national mission in certain key areas. There's great opportunity in areas like nanomedicine, synthetic biology, that we should, very intentionally take a leadership role in. And that comes with the government's intent, that is that they will invest in these areas, and that is invest for the long haul. And that type of a strategy needs to be resilient to the government of the day, right? So I think that would be my message. And in that, I think all of us have a role to play, because as a scientific community, the more we are able to collaborate and innovate and identify those grand challenges that Canada needs to address, the more easy it will be to convince the government of the day, and more difficult it will be for any government coming in to say this is not a priority area. So I think we have to have that clarity of thought as to collectively what are the 1 or 2 or 3 areas that Canada wants to be a leader in and invest in.
Yes, thank you. I think when we have a viral attack, I don't think it recognizes who we have in the government. So it hits everybody the same way. So that is true. Dr. Jeffs?
Yeah, I agree with all of that. And I think also is that we have to think about our emerging talent, developing talent in Canada. I agree with your earlier statements about keeping that talent in Canada and letting them develop, providing them the skills and the training so they can move into other parts of pharmaceutical development. From the bench, I started as a bench scientist, as probably many of you did, and ended up manufacturing GMP investigational drugs. So how did I get there? And I had some great mentors. But at the same time, we have to formalize that training so we can grow our industry.
I think we have learned a lesson this time, and I hope that we know what to do next. Dr. Racine?
Yeah, I echo all those comments that, and I think what I really want to emphasize, to the governments of the day and future governments is, don't forget the lesson that we've learned this past year. In infectious disease in particular, because that's my area, and so I see it all the time, is there's an outbreak of something, and we focus on it. They hand out a lot of money, have great intentions, and then that outbreak dies away, and then we forget, and then it happens all over again, and then we forget. I mean, probably this one won't be as easily forgotten because of its magnitude and duration. However, please, let's not forget that we need to continue investing in research and development, in the creation of new platforms, new technologies. We need to keep manufacturing capacity, supply chain, capacity here in Canada, so that we're not so reliant on outside countries, organizations for protecting our own people. And so hopefully that is something that the government will remember, and not just this government, but all future governments.
I mean, we have the great minds here. There is no way that we can't have a vaccine from start to finish made in Canada. And absolutely true. I think I can't agree more with you all. So thank you, doctors, Jeff, Krishnan, and Racine. So I think we have the last 5 minutes for some questions, and I'll open the floor for some questions. Marshall, do we have any questions?
We do. We've got a raised hand and a few written questions. So I'll take them in order. The first person to indicate the question was Mustafa Gozlan. If you want to unmute yourself, Mostafa, you should be able to speak now. Go ahead.
Yeah, sure. Everyone hears me?
Yes.
Fantastic. Thank you so much for that fantastic presentation from all the panelists. From a technology transfer perspective, I had a concern, and any of the panelists could answer me kindly. How do you see that intellectual property rights to inventors and research centers and universities and so on and so forth, in addition to financial profit that is always or oftentimes an interest of companies, how could those two factors be an obstruction to a possible powerful collaboration, allowing all those Canadian institutions and companies and startups across the country to reach full potential to nanotechnology in general and to vaccination in particular, in specific?
Who would like to answer?
Well, I guess I could take a first stab at it. So yes, absolutely. There could be potential roadblocks in, moving products forward because of the desire for maintaining IP or, having the biggest profit margin. But, I think that's something that groups need to work on together to identify pathways that works for everybody. You have to recognize potentially where your source of funding is coming from. If you've been funded by the federal or provincial government, well, then you should not, in my opinion, my personal opinion, not be so concerned with that big profit margin because, it's come from taxpayer dollars. And so hopefully, the groups can work out an agreement that would benefit everyone so that that isn't a roadblock. And especially, during a significant outbreak or a pandemic, profit should not be the key. And we have to keep in mind that while you may not make as big of a dollar on a particular product during a certain situation, if that has spurred a whole lot of innovation and new discovery, that could translate down the road to new products as well that would, potentially generate some profit as well.
So if I might add to that, I think there was two things in any type of innovation, right? You want to look at from the outset, coming together in any collaboration, you want to define what's your background intellectual property and what's any arising intellectual property where you actually collaborate and contribute and co-own and there's a co-inventorship. And this doesn't matter whether it's open science or whether it is going to be patented. The principles remain the same, that you just need to have a very clear understanding of who brings what to the table and how together you're going to get somewhere. And then there's many different models. Once you kind of clarify that, there's many different models you can work with, because you have to realize Canada and the market opportunity in Canada is only 2 to 4% of the world market. Mm-hmm. So there is easily an opportunity to, depending upon where your funding sources are coming from, to arrive at agreements where you get preferential opportunity to bring something. So if the government invested a lot and de-risked a lot, maybe there is an offset of that pricing for Canada, but you still have the world to take your product too. So there's many, many different ways. There is opportunity to look at, how do you pool your IP and move it together? But in order to any of these business models to work, you have to come in with a very clear understanding is what is your background that you bring to the table and what are you going to together have as a rising coming out of that innovation. And I think those conversations and negotiations then frame the possibility, and there's a lot of ways in which you can bring those aspects together.
Thank you, Dr. Krishna. Maybe one more question, Marshall?
But I'll read one out now, and it's from Thomas Skrinskas, who asks Dr. Jeffs, are the manufacturing capabilities going to be able to accommodate other APIs outside of RNAs? For example, insoluble drugs that require some sort of nano delivery platform?
Definitely, yes. I think that's the versatility of the nanomedicines, nanoparticles, highly versatile for genetic medicines, RNAs, DNA, and also small molecule drugs and drugs that are sparingly soluble as well. So yes, most definitely.
Dr. Jeffs, my question is, do you work with cannabis as well? With your technology, would it work with any cannabis products?
Yeah, I think it is compatible, definitely. Yes, it's not something that I've worked on personally, but there are local companies in the Vancouver area, one in particular, that is looking into that quite actively.
I may come back to you on that later. I think we are, I think our time is already, we are up. And so I would like to thank Dr. Jeff, Krishnan, and Racine. And please join me in thanking all of you for participating in the first panel discussion of the day. Thank you.
Thank you very much, Nuzat, and our wonderful panelists. I think that was a fascinating discussion, and I thank you all for participating. Now it's my pleasure to introduce the second keynote speaker today, Dr. Thomas Madden. Dr. Madden is President and Chief Executive Officer of Acuitas Therapeutics, and he founded the company in 2009, and he's guided it to a leadership position in the application of lipid nanoparticle, or LNP technology, for the delivery of nucleic acid therapeutics. Now, recently, Acuitas has focused on the development of LNP carriers for messenger RNA therapeutics. And this includes the LNP carriers that are used in several COVID-19 vaccines from the Pfizer, BioNTech, CureVac, and the Imperial College London products. So it's my pleasure to invite Dr. Madden to share his talk, Supporting COVID-19 Vaccines and Perspective on Future Canadian Pandemic Preparedness. Dr. Madden.
Thank you, Marie. I will just share my screen now, if that's okay. Hopefully everybody can see this. So first of all, my thanks to the organizers for the invitation to present today. It's a pleasure to be joining you all. And certainly I want to point out that my presentation is really set at a 50,000-foot level. I deliberately sort of stayed away from, some of the scientific and technical aspects, and I really want to talk a little bit more about some of the sort of fundamental learnings I think we have from the experience we've had going through COVID-19 and how those have influenced our thinking on future pandemic preparedness. One of the things I think it's really sort of interesting to look back on is that when first reports of COVID-19 came out, that was in December 2019, and by January 10th, the virus had been isolated and sequenced, and we knew from that sequence then the protein components that the virus encoded. And that's a remarkably short period of time in order for that information to become globally available. And I think it was the foundation for many of the vaccine efforts that then kicked into gear, looking at how can we develop a response, a vaccine response against COVID-19. Early studies quickly identified the role of the spike protein, as in other coronaviruses, in facilitating infection of human cells via its interaction with the ACE2 receptor. And so, of course, it was very immediately appreciated that it would be an excellent target for a protective vaccine. And as you're aware, it has been, I'd say, virtually the sole target that people have looked at. I wanted to sort of quickly talk a little bit about the concept of using messenger RNA in a vaccine therapeutic or a vaccine product. Clearly, a synthetic mRNA can encode any protein we want. In this case, the decision obviously was to use a message to encode the COVID-19 spike protein, with the idea if we can then have that protein expressed within our cells, we can trigger an adaptive immune response that will protect people against future infection by the virus. And it's obviously been known for quite some time that the major challenge around messenger RNA vaccines is that the mRNA is highly labile by itself after it's administered into the body. RNases chew it up remarkably quickly. And as a huge, highly charged molecule, it also can't get into cells where it needs to be in order to be translated. And so the challenge obviously was to identify a delivery platform that could protect the mRNA and get it into cells. And I'm just going to share hopefully a video. I can't seem to share the video. I don't know if that's the platform is preventing me. Sorry. Oh, here we go. So here we show the mRNA protected inside its LNP carrier, binding to the cell surface, and then the carrier enables escape of the mRNA into the cytoplasm where it can be translated into the new protein. And if I can now just close this. Oops. I have too many things preventing me from closing this here. Okay, so as I say, the solution to enabling messenger RNA vaccines was really around provision of a delivery technology that protects and delivers the mRNA. And it's sort of interesting when we sort of look back at COVID-19, it's important to recognize that Acuitas had been working with our partners on development of mRNA vaccines prior to COVID-19 coming around. And in fact, we had been working on a rabies vaccine with one of our partners, CureVac, in 2019. And the phase 1 data for that vaccine were released just in January 2020, showing that we could get a very powerful protective immune response at a remarkably low dose of the vaccine, 1 microgram mRNA, practically a homeopathic dose, as I like to refer to it. So that early clinical data, exciting early clinical data came out just as the threat from COVID-19 was becoming apparent. And so we quickly engaged with our partner CureVac and our partner BioNTech and decided to employ this same carrier system, this same technology, to now develop a vaccine against COVID-19. And fortunately, that effort was successful. And it's remarkable to look at how quickly we actually were able to advance a product into the clinic and have it approved for emergency use. And so the BioNTech Pfizer started their clinical studies in April 2020. And I think what was really sort of innovative is that they decided to take 4 different candidate vaccines into phase 1/2 in order to be able to make an early decision as to which seemed to have the most promise. And I think that clinical innovation, I think, was really instrumental in ensuring that the best candidate moved forward into large-scale phase 2 and phase 3 studies. BioNTech Pfizer initiated their pivotal phase 2/3 study in July 2020, remarkably soon after the phase 1 studies, and the results, the initial positive phase 3 results, were released in November of last year, allowing authorization for emergency use in December 2020. So a remarkably fast development timeline, while at the same time ensuring that all of the necessary clinical data to confirm the efficacy and safety of the vaccine were generated. I wanted to switch gears a little bit and just talk maybe about some of the challenges that were faced, in order to be able to make available the billions of vaccine doses that are needed once the clinical data confirm the efficacy and safety of the vaccine. And I think it's worthwhile sort of looking back at the quantities of components that are required for an early stage clinical study relative to the quantities needed for large scale commercial distribution. So for a phase 1 clinical study, 5 grams of mRNA would be more than adequate, and it's that sort of quantity of mRNA that companies were focused on producing in order to support clinical development of their mRNA products. That then needs to be scaled up to in excess of 450 kilograms of mRNA in order to provide 2 billion doses this year. So that's a phenomenal scale up. And it's equally impressive when you look at the lipid components. So for a 5 gram phase 1 batch, you need maybe 100 grams of lipid. To provide 2 billion doses, you need in excess of 9,000 kilograms. And so if you hear anybody complaining that we hadn't scaled up manufacturing of this vaccine fast enough, then by all means give them a kick in the shin for me, because I really think it's been a remarkable effort and one that continues to look at how can we further improve manufacturing capacity. Similarly, in terms of the vaccine product itself, from needing somewhere in the range of less than 100 vials for a phase 1 study, we need 330 million vials for 2021 commercial supplies. And we talked earlier about the challenge just in getting that many glass vials. I mean, that is a phenomenal number of glass vials you need above and beyond what the world manufacturing expectations were for 2021. So as I say, I think we've done a remarkably good job, considering the magnitude of the challenge. I also wanted to reflect a little bit on the foundations for success, and it's important to recognize that this success was built on strong pre-existing relationships we had with our international partners. We'd been working with CureVac and BioNTech in particular for several years in advance. We also had strong relationships with the CMOs that provided our lipid components, our proprietary lipid components under CGMP manufacturing conditions, as well as manufacturing finished products. And so as a result of those relationships, we were able to engage at the highest level with those companies, so on a sort of president to president basis, and ensure that we got that commitment to this development effort right from the start, back in February of last year. The other thing that's been important for success is parallel outsourcing, because no one manufacturing site, no one CMO is able to provide sufficient supplies to meet global needs. And so there's been a phenomenally efficient effort in engaging additional partners and having them employ established processes in order to be able to support the manufacturing efforts. I wanted to also sort of quickly talk about the emergence of COVID-19 variants. And as I'm sure you're all aware, these variants represent amino acid changes within the spike glycoprotein. And these variants can make the virus more easily transmitted. We've certainly seen examples of that, and potentially they can also trigger more serious disease. And so, they are obviously a concern. And the more widely COVID-19 is spreading throughout the globe, the more rapidly variants will emerge over time. What about how do we address variants of this virus using an mRNA vaccine? So obviously the current mRNA vaccines express the original Wuhan spike protein, and the antibodies generated by patients who have been vaccinated using, for example, the BioNTech Pfizer vaccine appear to be effective against certainly the UK and South African variants, but with a lower neutralization efficiency. And so there's obviously the concern that variants or combinations of variants could arise that escape protection by the current vaccines. One of the beauties of the mRNA vaccine approach is that it would be relatively trivial to revise the mRNA sequence to exactly express a new variant spike protein, or alternatively to include multiple different mRNA sequences in a single vaccine to express both the parent original protein and one or more of the variant sequences. So I think this technology lends itself very well to being able to address variants as and when needed. What's also been helpful, I think, is that the regulatory agencies have quickly engaged to consider what type of clinical studies would be required in order to approve emergency use of variants of the current vaccine. And guidance documents have been released both by the European Medicines Association and the FDA. And these guidance documents assume that a variant vaccine would use the same technology and the same platform as the parent vaccine. As I say, that's straightforward, obviously, for an mRNA vaccine. And what's encouraging is that the regulatory agencies are recognizing that large-scale clinical studies are neither required nor appropriate. Delaying provision of a variant vaccine in order to conduct a phase 3 study, for example, would be counterproductive. And so what they are recommending is that clinical studies for a variant vaccine should really focus on comparing the parent and the variant in terms of the immunological response they generate, and presumably also the neutralizing ability against the variant strain of the virus. Both BioNTech, Pfizer, and Moderna have already announced plans to evaluate what they're referring to as booster vaccine administrations, and these could represent a variant vaccine. But to my knowledge, the intention from both BioNTech, Pfizer, and Moderna is that the variant vaccine would be used as a booster rather than to replace the parent vaccine, and that's important in terms of the regulatory consideration that's required. I want to quickly sort of switch and talk a little bit about future pandemic preparedness and sort of vaccine requirements there. So clearly, we want a clinically proven vaccine modality. We want we want the ability to rapidly take a clinical candidate through development. We need a scalable manufacturing process using proven technology, and we need clear clinical and regulatory guidances. In terms of the vaccine modality we should be considering for future preparedness, what I've shown here is the sort of efficacy data for the 4 vaccines that are now approved in Canada. And it's sort of interesting, the 2 mRNA vaccines have 94 and 95% efficacy, which is significantly higher than the AstraZeneca viral vector or the J&J subunit vaccine. And I'm possibly biased, but I would suggest that if we were to want to place a bet on a vaccine modality to address a future viral threat, I think going with the mRNA vaccine approach would be the wisest course. And so in subsequent slides, I'm kind of assuming that that's at least the preferred modality, and then looking at what we would want to do in order to be prepared for future pandemics. As I say, the advantage of the messenger RNA vaccine is that it builds on our ability to rapidly isolate and sequence a viral threat, and then quickly generate a message to encode a viral component that would appear to be a promising candidate for neutralizing a target. The other beauty of the mRNA vaccine approach is essentially a plug and play, where you can simply substitute in a new mRNA into the same LNP carrier system. You can keep all of the components, the excipients in the vaccine the same, but now you're delivering a messenger RNA that encodes against this new viral protein. And so you already, if you've done that, you already know what are the lipid components you require, what's the manufacturing process. You could pre-select what's the product format size in terms of vial size or syringe size, fill volume, everything else based on the experience we've already generated with the current COVID-19 messenger RNA vaccines. The other advantage for this approach is that these are really highly effective and efficient vaccines. For example, certainly for the CureVac and BioNTech vaccine candidates, the dose is somewhere in the 10 to 30 micrograms messenger RNA. And so if we were looking at providing sufficient doses to provide 80 million doses to inoculate everybody in Canada with 2 injections, then that's only somewhere in the region of 1 to 2.5 kilograms of messenger RNA. So it's a very realistic quantity of payload that we would need to be able to manufacture. The other thing, a couple of other things that we've learnt, and it's already been commented on, is what have we learned from COVID-19? As I mentioned earlier, the idea of taking multiple vaccine candidates into early clinical evaluation is, I think, a very good one. I think one we should consider for future cases, because it allows us to maximize safety and efficacy prior to starting large-scale phase 3 trials. The other thing that has already been commented on, as I mentioned, is the challenges around having access to the vials and the syringes and the components, the lipid components, etc., that we need in order to manufacture. But if you go with a messenger RNA plug-and-play vaccine candidate, then you can stockpile all of these. You could stockpile the nucleotide bases you need to synthesize the mRNA. You could stockpile all of the lipid components, the vials, the syringes, etc. What we would need to do, though, is identify the manufacturing facilities, both for the manufacturer of the mRNA drug substance, as well as the fill finish facilities to manufacture the final vaccine itself. And that's, as I say, simply noted on this slide here. And I think one of the in my mind, one of the key areas where we are sorely lacking is in a standalone facility that has the expertise to generate messenger RNA, both for academic research, commercial research, but also commercial production. That's certainly a capacity that's lacking within Canada. With respect to some of the other components, as I say, we don't necessarily need to produce them in Canada if we've already pre-selected what they are, and we simply store them in Canada for use in the case of a future threat. So I wanted to just thank all of my colleagues at Acuitas Therapeutics who did all of the work, obviously, in supporting our partners, and also wanted to thank our colleagues at BioNTech, Pfizer, and CureVac, who we've worked with very closely and who've done a fantastic job in bringing these vaccines to the market, or in the case of CureVac, very close to the market. So thank you.
Thank you very much, Dr. Madden. That was fascinating. Now we can take a few questions. So let's see. Marshall, do we have questions? You can either raise your hand and Marshall will open your camera.
No hands at the moment, and no written questions yet.
Okay. Maybe I have a question in the meantime. So how long had you been developing this technology? I mean, of course you were ready in a sense to act very quickly when COVID-19 hit, and it's quite amazing that all of that work could be done so quickly. But you know, how many years have you been building up, in a sense, the technology to be able to respond?
Well, I mean, the use of lipid delivery systems for drugs has been something that Peter Cullis in his group initiated back in the 80s. So literally decades, but obviously the systems have evolved substantially during that period. Within Acuitas, around 2012, 2013, we recognized that people were talking about messenger RNA potentially being a new therapeutic. And we recognized back then that the key challenge would be delivery. And so it was then that we sort of refocused our efforts within the company to enable delivery of messenger RNA. And we were very fortunate to set up some early collaborations with key figures in the field, Dr. Drew Weissman, for example, Katie Carico as well. And with them and with some of our early industry partners, demonstrate the utility of LNP systems for mRNA delivery. But we'd been working with CureVac and BioNTech for several years prior to engaging on development of the COVID-19 vaccine.
Thank you. I think we have a question in the Q&A.
We have, and a raised hand. I'll start with a written question. Luis Perez Davalos asks, with the lessons that we have learned developing the SARS-CoV-2 vaccine, would it be worthwhile to try to make mRNA alternative vaccines for the existent immunizations based on inactivated or attenuated microorganisms, fragments, etc.?
I would say a definite yes. I mean, I think, I mean, COVID-19 mRNA vaccines are the first approved vaccines, and I think they've really demonstrated the power of this approach. And clearly now, I think everybody should be looking at how else can we apply this technology. You know, from flu to SARS-CoV-1, but also to some of the more intransigent challenges such as HIV and malaria. And again, those are both areas that we are heavily working with academic researchers, and we would love to see now clinical studies initiated to expand, the potential application of mRNA vaccines.
Great. If there's time, Nusat has her hand up. Please go ahead.
Hi, Dr. Madden. It was a fascinating presentation. So my question is, of course, we all would love to take the mRNA vaccine, looking at the efficacy, but do you think that we should wait for mRNA, or should we just get whatever is available right now, which is the AstraZeneca?
Well, I'm clearly biased. If I had a choice, I'd go with the BioNTech Pfizer vaccine. But I totally agree. The best vaccine is the one you're offered. And I think we should definitely continue to promote that message.
Thank you.
Thank you very much, Dr. Madden, for such an inspiring keynote address. Thank you.
Thank you.
So now I'm pleased to introduce two additional panelists who will join me for the Canada's Nanomedicine Cluster funding announcement. Monsieur Stéphane Gagné has 25 years of experience in the fields of cosmetics, nutritional supplements, pharmaceutical products, and active ingredients. Since 2006, he co-founded 3 companies where he held the role of president and CEO. Mr. Gagné is the co-founder and president and CEO of Ovensa Incorporated, a nanomedicine company that has developed an siRNA targeting galectin-1 to fight resistance in head and neck cancer. Ms. Rhonda Gosselin has spent most of her career as a technology advisor focused on life sciences, first with the Genesis Center at Memorial University, and then at the National Research Council Industrial Research Assistance Program, or NRC-IRAP, for the last 20 plus years. And in her current role as International Industrial Technology Advisor and National Program Lead for the Canada International Innovation Program with Brazil, she's helping Canadian SMEs to connect into global value chains through co-innovation relationships. Welcome, Rhonda and Stéphane. And I will share my screen. Okay, so hopefully you can see my screen. Yes?
Yep, looking good.
Okay, good. So, all right, I will start then, and we'll talk about investing in Canada's nanomedicine ecosystem. And you might first ask, well, what's NanoCanada anyway? So NanoCanada is a not-for-profit corporation, and we were founded in 2015 to address a specific need, and that was to connect academic researchers with industry. And we felt there was a window of opportunity. It was important to do that at that stage of development of the nanotechnology. And so what we've been doing over the years is really being the connectors. We get a lot of calls from companies that say, I'm looking for a partner in this particular area. Do you know of anyone? And we can connect the right people, the right expertise, give access to state-of-the-art facilities. And this is really for the advanced materials and nanotechnology community. So we are, of course, membership-based, and a lot of our members are small and medium-sized enterprises, some academics, and also we have government partners. But more importantly, we represent all of the hubs across the country that exist so that we can speak with one voice when we go outside of Canada. And I think that's extremely important. And some of the work that we do is actually hold events, both in Canada, but abroad. And we have turnkey missions that help Canadian companies bring their products to global markets. And so my next slide shows some of the opportunities that we offer to our members. Is We will be organizing our first international conference in 2022, but we have a launch event in 2021. And I'm showing this because amongst the themes is something related to health and nanomedicine. So we can choose the events to highlight some of the applications of advanced materials and nanotechnology. And I think that's really the power of having a strong ecosystem, so that we can tell the story and we can show how these technologies, whether they're emergent or well established, weave together to advance innovation in Canada. And we've just heard that story in nanomedicine, and how important it is for manufacturing, for example, to work hand in hand with those who develop innovative ways of delivering vaccines, for example. So I will let Rhonda speak to the next slide.
Well, thank you very much, and thank you for inviting NRC to present. So just highlight very quickly before we move into the rest of the presentation. The National Research Council is mandated, it's 100 years of experience. It's the largest research org within the Government of Canada, focusing on innovation and research with a commitment to business innovation, supporting policy solutions for government, the science agenda, and advancing knowledge through science. And we do that, and the commitment is there through the NRC Research Division, which involves innovation for the benefit of Canadians. And what NRC has done is a platform, a partnering platform for innovation across the country to help accomplish that with its 2,000 plus research scientists. And I just want to highlight the program, the research program is divided into 5 research divisions, 14 research programs, and one of those is on nanotechnology. The second aspect of how NRC helps accelerate growth and innovation in this country is through supporting SMEs. And that's the organization with which I work. I've been there for 22 years, and we have 255 plus industrial technology advisors across the country, helping SMEs to innovate. And at that point, I'll pass it over to Stefan.
Sorry.
Thank you, Rhonda, and merci, Marie, and thank you very much for the invitation. Good afternoon, everyone, and good evening if you're following us or watching us from Europe. Again, I'm Stefan Gagné. I'm the president and CEO of Ovensa, based in Ontario, and Ovensa Innovations that we incorporated last year in the province of Quebec. Just before I share with you a little bit on the reasons and why I believe very much in the announcements for today, and this idea of why we need a nanomedicine cluster, I'll give you a little bit of news about Ovensa, information on what we play and what we do in the field of nanomedicine. So in short, we use a biopolymer that we call triazan, and to make nanoparticles that will carry one or several molecules. We've done even different ratios of molecules, of small molecules within the same nanoparticle, and we can bring them to targeted tissues or cells. So the cationic charges on the surface of triazan help us to cross the barriers, the tight junctions, or the blood-brain barrier. I would say that most of what we do today is with pharma partners is to cross the BBB. But even to be more specific to certain types of cells, or tissues, we can conjugate ligands, what you see on the surface there of the triazan, ligands that would be specific to certain receptors on the surface of cells, so that we can bring those, the cargo basically, more specifically toward certain tissues. And as Marie explained in the introduction, and I'm thankful for that introduction, Marie. We are developing our own drugs, which is a siRNA targeting galectin-1. So galectin-1 is a protein that is very much involved in cancer resistance, kind of a troublemaker. So not only we are developing our own drug candidate in head and neck cancer, but also we collaborate with the industry. We collaborate today, we have 6 collaborations with big pharma. Most of our work is in the RNA space, so siRNAs, mRNAs. And I cannot only think of the potential collaborations when I've listened to the different presentations today, the researchers from the academy, on how we can further collaborate, and even with NRC in that presentation, how can we better collaborate in Canada today? So next slide, yeah. Basically, how did we come up with this idea of a nanomedicine cluster? How did that started? Well, NanoCanada had organized a commercial mission in Brazil where Ovensa participated, and I met with various pharma companies, nanomedicine companies, and it's when I met with a company, pharma company in Brazil called Aché and Ferring, a Switzerland company based in Switzerland, but having offices and research facilities in Brazil, that I learned about their creation of a nanotechnology innovation lab to do basically exactly what Ovensa does. So they call it NILE. And what we do and what they were trying to do, and they announced, was they tried to form nanoparticles for specific targeted drug delivery purposes. So I thought, why not collaborating with them instead for them to try to reinvent the wheel, basically. So precision nanosystems that we heard presentation earlier today were also part of the mission. So when I met with Samuel and looking at what we could do together, how we could partner to offer basically a combined solution with using their microfluidic equipment and our polymer platforms, then I thought, let's do something and propose that. So during a presentation at the Chamber of Commerce, Brazil, Canada, Rhonda and I were sitting next to each other, and I shared those outcomes of the potential of teaming different groups within Canada so that we can provide a solution basically to Aché and Ferring. And basically she jumped on the idea. She said, why don't we do a cluster? NRC could support this. And we obviously thought that NanoCanada would be the best entity to lead this project. So that basically became the start of the Nanomedicine Consortium. And this week I look back at my notes taken at the time in that auditorium and on OneNote, and it showed me that it all started at 9:37 a.m. Brazil time on September 26th. So I put it up there, and I realized even this morning that that's exactly also when I joined this mission in Brazil. An anecdote here: the morning the day I was taking the flight, so my flight to Brazil was at 11 p.m. And in that same morning, I had a concussion around 8 o'clock, I think, in the morning. Went to the hospital. I asked the doctors if I was okay to go to take a long flight to Brazil. And basically, they said, well, we have nothing in your blood work to show that you shouldn't go, but that would be a little bit crazy. So crazy as I am, I decided to go. I joined the mission and did not regret it. And also, I think that coming back to the reason also why we're doing this and why the idea came up is that when I looked at the stats that were presented, that's why I put this picture of a slide that was presented at a time. There was a lot of money that was invested by the Brazilian government. So they've invested over $200 million Canadian, the equivalent of Canadian dollars, in 15 years in nanotechnology, right? And forming clusters and groups that would get together with complementary skills. And we thought, why can't we do this in Canada? I think that we can even do better than that. And that's exactly what we're doing today. We just need to get basically to get organized. Next slide, please. So what's in it for us, and why this nanomedicine cluster for our industry? Beyond what I just said, again, I'm always surprised to meet more of my Canadian fellows when I am outside of Canada. And we meet more often outside of the country than we do in the country. And what I like about this initiative is that we'll get to know and collaborate with each other in the country so that we can come as a stronger voice and be more competitive outside of the country. So that's why I put there the let's buy, build, and partner in Canada first. So just this thinking that we're having today with the different industries and how can we collaborate more. And following that mission in Brazil, well, Ovensa started working with two companies that were part of the group. So we decided to buy the microfluidic equipment from Precision Nanosystems. We started using the services of Ananda that was there. And by getting to know more companies from our ecosystem, I think that we can develop even more partnerships. With the building of a centralized database of our complementary expertise, and the promotion of our Nanomedicine Cluster, I think that we can all win together. And with NanoCanada leading this cluster, having the support of NGen and NRC, so powered by, I like to call it, I'm very confident that our nanomedicine industry in Canada will greatly benefit and make us collectively more competitive at the international level. So that's it for me. Thank you.
Thank you. So, I think some of you might ask, well, what do you include in nanomedicine? And we don't want to be exclusionary. And as I said before, the real opportunities that I see is that we have such a rich community in advanced materials and nanotechnology that can work hand in hand with companies and researchers who work on nanomedicines. I think we have a wonderful convergence that is a competitive advantage, as we've heard from the success stories of our presenters today. And so I don't want to spend a lot of time on that, but it is all about convergence and about collaboration and how we work together. And so that's the advantage of having a cluster. So I'm really so pleased to announce today that NanoCanada has received funding from Next Generation Manufacturing Supercluster to do this nanomedicine cluster build in order to better connect the community into a rich ecosystem. And we've heard how this has happened, the number, we've had conversations over the years, but certainly in the last year when we're talking to Peter Cullis at Enmin and Diana, and what are we missing? Because we're always thinking, what's the gap, and how could we do something about it? How can we leverage the community to act, to advocate, and see how we can find solutions? And I think we were certainly encouraged by NGen to put in a proposal as a starting point. It's only a starting point, but we have to start somewhere, and that is where we are starting. But it will lead to much bigger things, I hope, and all linking and building that advanced manufacturing capability in Canada, but with all of the other interlocking pieces, as we've heard today, in terms of supply chains. And so, as I said, we have a one-year plan that's been funded, and you see some of the deliverables there in terms of having a digital platform for the community, and of course we will continue with the other activities that we have nationally and internationally, and you're most welcome to join us in some of those. And really we'll be there to serve the community and showcase your success. That's what we do. And so that's what we hope is going to happen here is that we will put Canada on the map. I think Canada is already in the map, actually, nanomedicines, but the recognition as a global partner in this field, and we're going to be able to grow collaborations and increase our innovation and capabilities, And a well-networked nanomedicine industry linked to advanced manufacturing platforms will be a great asset to Canada. And I really am inviting you to join us in this adventure. And I will let Rhonda talk a little bit about what she sees as other things that could be leveraged from this build, and I'll come back with a call to action at the end. So Rhonda.
Sure, thank you, Marie. So as I alluded to earlier, the National Research Council, IRAP, has focused for 75 years on innovation and helping SMEs become more innovative. In recent years, only since 2013, the International IRAP program has started to actually formalize and look to accelerate the growth of SMEs through and groups of SMEs through innovation and through international partnerships. And what we've been doing in the last 7 years at the international group is developing an office that has put in place MOUs and the opportunity for SMEs to collaborate abroad in the spirit of co-innovation. And we have bilaterals and MOUs now that support collaboration with Germany, UK, Japan, as well as our companies now from Canada can participate in the Eureka framework, the European platform for doing research and development, and the other countries such as Canada that are partners to that. What's really interesting about that platform is that they also have 7 clusters, consortium clusters that are part of that framework. One of them, of course, being advanced manufacturing. So the international IRAP has really been focusing on helping companies accelerate their competitiveness and their growth by actually getting them to think global and to help insert them into global value chains. Next slide, please. And why is NRC IRAP doing that? And as you probably saw from my first slide, our role in the Government of Canada is to enhance the competitiveness of our SME community. And the results of the Eureka program, which has been around since 1986, it is the platform for collaboration amongst all of the European countries and of course its associated partners. And the results speak for itself. It truly, SMEs that go global, that co-innovate globally, are truly more competitive in all shapes and forms. Next slide, please. And so this is my last slide, and I think it's probably one of the, hopefully will provide the context for why I'm actually speaking and why NRC is at the table. Because NRC IRAP for 75 years has focused on the firm and the innovation in the firm in Canada. And in the last 7 years, NRC IRAP is focused on helping the firm connect to international consortiums. So you can see it's one end of the spectrum to the other, and helping our firms insert into consortiums and clusters. And when we were on the Brazil mission with Stefan, what we saw was, yes, 11 firms participated in the Brazilian mission. 5 of those firms found foreign partners for co-innovation, but what was really interesting and really stood out was that 11 domestic collaborations were noted as a result of being on the ground together for 5 days. And when they come back to Canada, they were going to start to collaborate. They didn't even know each other. And what we realized, and NRC IRAP has seen this in all of its international programs. So in 2020, in an effort to look at this and say, well, how do we support Canadian firms? One, to flow into super clusters within Canada, and how do we support our super clusters and leverage the IRA program? How do we continue to support our SMEs and getting them into these consortiums abroad and broaden their competitiveness? And we thought about piloting a Canadian collab program. And in 2020, we had 36 companies in the cybersecurity sector come together in a workshop. And of those 36 companies, we had 4 consortiums that were created and supported through IRAP. And what is really fantastic about this is that one of those consortiums actually went on to participate in the ITA3, which is an IT cluster within the Eureka framework. Furthermore, one of what's really, I think, fascinating about this whole IRAP cluster program was that 13 of the 18 firms across Canada that actually went abroad to participate in this ITA3 Eureka cluster framework came from the Canadian Cloud program. So even in our first pilot, it clearly demonstrates the value of domestic and supporting domestic collaborations, pulling clusters together, helping companies facilitate that interaction, supporting them in those efforts, and moving them along that continuum into the national ecosystems, which is the ICET program, our superclusters such as NGen, and of course then taking all of that and moving them which into international consortiums. And I just want to conclude to say that having a leader though, such as NanoCanada, having an organization take the lead to really pull all of this together, because this takes initiative, it takes structure, it takes organization, it takes knowledge of all of the various programs that Canada has to offer. And we have one of the richest, innovative R&D ecosystems in the world that supports R&D at all levels. And what we really need to do is be leveraging that. And we can only do that if we come together in the manner in which we're talking about today. And so I'll end there.
Thank you, Rhonda. So we hope then that, we will build this cluster. We'll have a very strengthened Canadian innovation network, and we have lots of partners to do that with, and we're very pleased about that. And really, the first thing that you can do is get involved, sign up for our newsletter, and you can contact us at info@nanocanada.com. And our cluster coordinator is going to be Isabelle Cyr. And actually, I'd like to invite the NanoCanada team, Janice Workington, Patricia Tokunaga, and Isabelle Cyr, if they're to open their cameras, actually, so we can see them. And so it will not be a disembodied voice at the end of the phone if you phone us. I don't know if that's possible. I'm not seeing
They
that.
won't have, they don't have the option for webcam,
Okay.
unfortunately.
Well, they're a great team, and you'll get to know them. So I'm so pleased that to have made this announcement today. Thank you, Stéphane and Rhonda, for your presentation and telling that story. And we have about 2 minutes for questions. So if there are questions for the 3 of us, we'd be delighted to answer them.
There is one written question has been submitted by Alicia Main, who says that this question is for Dr. Diorio. And the question is, have you tied into Canadian social science innovation ecosystem research on this topic? For example, the 4.0 Innovation Ecosystem National SHIRK Partnership. And if not, Alicia says she would be happy to help make the relevant connections.
Yes, Alicia, I look forward to working with you on that. I was actually just reading one of your papers on advanced materials. And so I'm very cognizant that this is incredibly important. And I think today we cannot do science without thinking of the greater social science aspects. And so having partners and keeping that dialogue going is really important. So yes, certainly let's get in touch and talk about how to weave that in.
And there are no other questions at the moment.
Okay, so thank you very much, Rhonda and Stéphane, for joining me in this announcement.
Thank you, Maggie.
Thank you. And now it's a great pleasure for me to introduce Peter Cullis, who will provide the third and final keynote talk today. And although Dr. Cullis likely needs no introduction to many of you, his credentials bear repeating. In addition to his role as NMIN Scientific Director, he also is a professor of biochemistry and molecular biology at the Faculty of Medicine at the University of British Columbia. He's been described as the grandfather of lipid nanoparticle technology, and he's been responsible for fundamental advances in the generation, loading, and targeting of liposomal systems for intravenous delivery of conventional and genetic drugs. This work has, of course, led to 4 drugs approved by regulatory agencies in the US, Canada, and Europe. And most recently, the lipid nanoparticle formulation, as we've heard, developed in his lab over 40 years, was licensed to Pfizer-BioNTech COVID-19 vaccines, and they've been approved around the world in 2020. Dr. Cullis has published over 340 scientific articles. He's an inventor on over 60 patents, and he's spun out 13 startup companies and received numerous awards, most recently the 2021 Controlled Release Society Founders Award, acknowledging his international recognition for outstanding contributions to the science and technology of controlled release. It's my pleasure to invite Dr. Cullis to share with us his talk titled The Importance of the Scientific Entrepreneur in the Translation of Academic Discoveries into the Commercial World. Peter.
Yeah, thanks very much, Marie. So yeah, well, I'm going to give a slightly different talk today. Really, it's an area where I don't think we have really tapped the talent pool as much as we might in Canada. We need to encourage more of our PIs in university settings to perhaps be a little bit more entrepreneurial, to try and get their discoveries that are made in the university setting out into the commercial world. And so I'm going to try and cover in the next 20 minutes or so, although I have too many slides, so I'll have to go a little quickly in places, is really why be a scientific entrepreneur? There's a lot of reasons for doing this that maybe are not immediately apparent at the outset. So I have a couple of conflicts of interest. I've co-founded Precision Nanosystems and Acuitas Therapeutics, 2 companies that are already presented today and are certainly in this presentation. Now, I am as Marie said, I am a scientific entrepreneur. I spent most of my career starting in the middle 80s, well, as my university professor career. I was appointed first in '78 at UBC, but I spent most of that career working half-time in academia and half-time in companies. And so this started with the Canadian Liposome Company in the middle 80s. I was the chief scientific officer of another startup that we founded as a result of the Canadian Liposome Company for 12 years, Inex Pharmaceuticals. And then heading up 3 not-for-profit companies that were really aimed at trying to get more of the bioscience discoveries into the more commercial arena. So this is the Center for Drug Research and Development, Personalized Medicine Initiative, and the Nanomedicines Innovation Network. Now, this was also paralleled by actually UBC didn't have to pay my salary until about, I don't know, the late 1990s as a result of all these other activities. Anyway, so I've co-founded 10 startups and 3 not-for-profits in the course of this time. So some of the points I want to make are, well, why be a scientific entrepreneur? And so there's a number of reasons. First of all, provide a benefit for the Canadian taxpayer. If you're a professor, create jobs for your students, and if you're a student, create your own job. Avoid writing grants. For those of us that are in the academic stream, this is a noble endeavor. They have fun, the most important part of all, actually, and then do new and exciting science. So I would say that, in answer to the question, why be a scientific entrepreneur, I'd say it's almost a moral duty if you're a professor Currently, more than 40% of our PhD graduates in science, technology, engineering, math, take jobs outside Canada. So we're really exporting a large proportion of our highly trained graduates, 350,000 or more Canadians in the Bay Area, another hundreds into the Boston area. It's probably larger than that now. This really doesn't benefit the Canadian taxpayer. It goes on. I mean, assuming an average cost for a PhD is $200,000 per graduate student, and 40% of our 5,000 PhDs that we grant in STEM sciences annually move south, we're paying $400 a year to support U.S. industry. This is not a good situation. Not only that, but we're supporting like wealth creation capabilities. Celera has a market cap that's north of 10 billion, but say it's 10 billion for round numbers. They employ 100 PhDs. They actually have about 250 employees. I would suspect about 100 of those are PhDs. There's a wealth creation of about 10 million per PhD. And so if the average PhD we export creates only a tenth of that, we're losing a couple of dollars a year in wealth creation and the tax base that implies. So again, not benefiting the Canadian taxpayer, who's really funding grants and our training programs. So this is I think a compelling reason why one should take a more entrepreneurial route. If you're a professor, create jobs for your students. If you're a student or a postdoc, create your own job. Another fairly obvious problem right now, we have only academic jobs. This goes to 2011, but the stats really haven't changed. I couldn't find the latest versions. There are only academic jobs for only 1 in 8 of our graduate students, postdocs. So we really need to find alternative approaches. Now, just to summarize a long story, we can create these jobs. So companies that I've been involved in co-founding now employ nearly 400 people in Vancouver. And we're a global innovation hub for nanomedicines. 6 of the 13 globally approved nanomedicines were developed in Vancouver. We have 4 companies employing over 300 people. And in the personalized medicine space, we have another 4 companies that employ some 75 people. So this is this is just to say that it's not pie in the sky to say that we can actually address some of these issues associated with the need to establish high technology industries. Avoid writing grants and being slightly facetious here, but this is something that university professors spend an inordinate amount of time doing, and it's a frustrating exercise. 15% funding, and we see enormous efforts going by very, very talented people to a pot of money that's clearly not increasing, hasn't increased over the last 10 years, and so the competition gets more and more severe. There's a lot more money in the available through writing business plans than writing grants. This is just saying 1 to 2% is in the grant system. There's the large majority is in the rest of the rest of society. And I would say a business plan is no harder to write than a grant. You just have to add some economic justification. Now on to the fourth reason for doing this: have fun. And this is an essential part of the commercial entrepreneurial and commercialization endeavor, at least in my view. So back in '78, we first formed the group. About 1985, we formed Lipex Biomembranes. So I was going to talk about the Lipex Biomembranes story, which then merged with another company we founded, Northern Lipids, in 2000, and then it was sold actually to Evonik, a large multinational firm, in about 2016. So in 1984, we'd invented a device for making unimolar liposomes. This was basic work in the lab. We called this device the extruder. And we formed a company called Lipex Biomembranes to market this thing, and we knew that companies had to have a mission statement. So we developed our own mission statement. This was free beer every Friday and parties whenever possible. It's still one of the best mission statements I've ever heard. So this is the device. We have lipids dispersed in water, and we push them through a polycarbonate filter to form these small unilamellar vesicles. Relatively simple process, and but producing really well-defined systems. These are the vesicles imaged by cryo-electron microscopy, and it's now the most favorite technique in the world. Certainly in every lab that is working in the area of membranes, you're going to find an extruder. And of course, when they progress through to clinical use, these lipid-based systems, then you need much larger systems. And there's now commercial instrumentation that I think is selling for about $1 million for each apparatus. Certainly free beer every Friday forever is the basic message here. So this is, we formed the company from, with me and four postdocs who were in the lab at the time. We were clearly having a good time doing this. So we made a decision at the board level to really that every serious company should support the arts. So we formed the Royal Lipex Ballet and performed unforgettable performances, which some of us would occasionally like to forget about, but unforgettable parties. You'll notice that this is the CEO of Acuitas Therapeutics who's taking a lead role in this. So I was taking this fairly lightly, but it's actually pretty serious. A serious success, certainly. We had no intellectual property. We had a good idea, had a reputation, and that tremendous amount of success came out of just moving into the entrepreneurial arena. Subsequent to merging it with Northern Lipids, as I mentioned, it was then sold. Lipex was then sold, or Northern Lipids was then sold to Evonik. And there's now over 100 employees in Vancouver. It's expanding very rapidly at the moment. So you can certainly have fun doing these kinds of endeavors. Another really, this cannot be underestimated or overestimated. You do have the opportunity to do new and exciting science. And so this evolved through three companies, Canadian Liposome Company, INEX, Acuitas, over the years. First of all, dealing with anti-cancer drugs, and then subsequently dealing with what turned into gene therapies. So we really, the initial efforts in the early 80s, middle 90s, and actually we still have it ongoing in the laboratory. It's just aimed at the ridiculous situation we have with cancer drugs, where we administer them systemically. Only 0.001% gets through to where the tumor is. The rest of it causes nasty side effects everywhere else. And so we need to get these drugs much more specifically to where they need to go. I mean, it's just one of those blatantly obvious problems that has not yet been beaten, but we're still going hard at it. But we made a number of advances that's really stemmed from quite basic research, actually. Methylamine, we're using it to measure pH gradients, and it goes across the neutral form. Getting into a little bit of science here, but the end result is that we have very high gradients of this methylamine marker that go according to the pH gradient. So it ended up for a 3-unit pH gradient. We can get 1,000 times higher concentration inside these vesicles. So this, from a basic science point of view, we then noticed that many anti-cancer drugs were weak bases. And we could use exactly the same procedure with a pH gradient to load in drugs to the point where they crystallize out on the inside. And we had remarkably good delivery systems to help us get these drugs more specifically to where they needed to go. So this resulted in a couple of drugs that we've developed with the Canadian Liposome Company and the parent company, the Liposome Company, which is based in New Jersey, that were approved in the US and Canada in 95 and 2000. The ribosome company, the parent company, closed down our operation in, this is some of the things that, some of the little bumps in the road, but actually it's turned out to be of great benefit. So we knew what to do at this point, so we were able to form our own company, which is Inex Pharmaceuticals. But again, you hit things, and when you're in the commercial arena, that you may not, if you stick with a straight academic career, you wouldn't have encountered. And this was, we couldn't raise any money and use it for delivering our cancer drugs in these lipid nanoparticles or liposomal systems. So one day the CEO told me, look, we can't just put old drugs in these carriers. We need to be doing gene therapy. It has much more sex appeal. So I wasn't too keen about this because we'd invested a huge effort in developing the systems to deliver drugs such as doxorubicin. And now we had to deliver much larger drugs like small siRNA, small interfering RNA. The first thing was that we had to find a way of encapsulating them, and all the agents that were available were quite toxic. We'd positively charged lipids, as you can see here, to get something that might pack into a hydrophobic environment. And these lipids they tend to rip membranes apart. And so what we developed was these ionizable lipids. And this turned out to be a huge breakthrough. Essentially lipids that have a pKa in the region of about 6.5. And so they're positively charged or protonated at a low pH, where you can do the encapsulation, and then you can bring the systems back up to neutral pH, and they retain the cargo. We also developed a rapid mixing process that Precision Nanosystems has really refined to encapsulate the oligonucleotides, the RNA, in these systems using this rapid mixing approach, where we have the lipid in ethanol, and we have the oligonucleotide in water, mix it very rapidly, and then we can develop these kinds of systems where we can more or less dial the size according to the surface to core lipid ratio. So we invented a new class of lipid nanoparticles as a result of this stimulation by our CEO to say, look, we got to work on some different systems here. And they had this had all the characteristics that one needs in order to be able to go forward with a system to test in vivo. One day I was at a conference in London, and I was accosted by the Vice President of Research for Alnylam Pharmaceuticals, a company based in Boston, saying we have a delivery. Alnylam is concerned as the lead company in the world actually delivering siRNA. And at that point, they said, we have a delivery problem. How do we get our siRNA into hepatocytes in vivo? And so using some of the systems that we developed, we set up a collaboration between my lab, Inex and Acuitas subsequently. And this was a huge effort for 7 years over the period 2005 to 2012. That really resulted in remarkable changes in potency for silencing a particular gene in hepatocytes. It then led to because we can silence genes in hepatocytes using this siRNA-based system, it led to a drug to treat a condition known as transthyretin-induced amyloidosis. It was introduced in clinical trials in 2012. And so this amyloidosis is caused by production of a mutant transthyretin protein in the liver. It produces fibrils in the circulation. These cause cardiotoxicity and neurotoxicity. As you can see, the progression is particularly nasty. There's about 50,000 people in the world that suffer from this disorder. So the hypothesis here was to deliver the siRNA into hepatocytes, silence the gene, reduce the circulating fibrils, and then perhaps reduce the deposition, maybe reverse it. And so this turned out to be one of the most successful clinical trial that I've ever seen. This is the neural impairment score that's basically over the 18-month course of the clinical trial. improved dramatically, a p-value that's is I like to say 1 over Avogadro's number. There is no doubt that this drug works. Quality of life, self-reported muscle strength. I mean, basically, we're taking this is an essentially curative disease for a previous therapy for a previously fatal disease. This was approved very rapidly by the FDA in 2018, but it points out the benefits of coming at something from a basic science point of view and being pushed into it in some extent by the necessities of the marketplace. So this went just to complete the story, going on to Acuitas Therapeutics, which has been mainly focused on messenger RNA gene therapies. We asked the question whether these systems could be applied to much larger messenger RNAs compared to siRNA. We found that the same encapsulation procedures could be used, and that we would see expression in the liver of the protein that the mRNA is coding for when we injected these systems intravenously. So these were then optimized to get maximum expression. But around about, this was a thing from 2012 onwards, around about 2014, we were approached by Drew Weissman at the University of Pennsylvania, who worked with Catalina Carrico. You've been concerned with trying to develop mRNA vaccines. So the same as Victor Taliansky coming to us, what Drew wanted, said, we have a delivery problem. How do we get mRNA coding for these viral proteins into muscle and immune cells in vivo? And so this was this required packaging the mRNA and then injecting intramuscularly to try and get a vibrant immune response, both from transfection of muscle cells as well as antigen-presenting cells, MHC1, MHC2 class responses to get a really comprehensive immune response to a given a messenger RNA that's coding for a particular viral protein. So this was applied, as Tom Madden said, to SARS-CoV-2 virus beginning in 2020, and this is the press release that was released in the middle of November in 2020, indicating that this vaccine was 95% effective. It was efficacious, independent of age, race, and gender. It was very safe, well tolerated, and that the manufacturing at that point was going to be 1.3 billion. They're now up to 2 billion. Obviously, this vaccine has been approved in many jurisdictions that we're well aware of. The same technology, as again Tom Madden was mentioning, can be applied to many other vaccine opportunities. There are also many therapeutics that are being enabled by this approach. So I think the importance of being a scientific entrepreneur is clear, and this is one example. It really needs to be encouraged. I think more definitely within the academic community, and taken advantage of, because you can do all these things that are indicated here, but actually turn academic discoveries into products that really help people. This is the result of a huge effort, and I should emphasize the team that originated this has been together for 40 years now. I worked for 40 years with Mick Hope and Tom Madden, 20 years with many others that are indicated here. And so the bottom line is you need a team that sticks together, and you need to establish as I mentioned, to have a hub or a center of excellence in this area. I think we've got that in Vancouver, but it takes a long time for that to develop in a really cohesive and permanent manner. Obviously, people at Alnylam, Marcus Saflini in the chemistry department, many others, Drew Weissman being pivotal amongst those in the areas of the vaccine. Anyway, so I think I've pretty much used up my time. So if there's anything left, I will use that to answer a few questions.
Thank you very much, Dr. Cullis. That was fascinating. Do we have questions, Marshall?
We have a raised hand. Nusat, if you wish to speak, go ahead.
Oh, hi, Dr. Cullis. It's really a fascinating presentation. Thanks for sharing your career with us. My question is, it takes a village to get one drug to the market, and we just heard that you have 4. What is your secret? Have you met with any roadblocks? If so, what were they? And how did you mitigate those?
Oh, yeah, well, you had all sorts of roadblocks, right? I guess the main thing is that you have to have, and this is where I think sometimes we don't quite recognize some of the issues, but you have to have people involved in making and pushing these things forward who are truly passionate about it and will not, whatever obstacle is just something to be surmounted, and not you don't give up. Because there's ample opportunities when you're trying to get a drug approved, or take a drug through the regulatory system, for things to go wrong, and we've had things go wrong. With Inex Pharmaceuticals, we had a drug before the FDA that we brought to the FDA, and it didn't get approved. And as a result of that, we had to go from a company that had 200 employees down to 50 overnight, and totally revamp and reorient the company to be able to move forward. So you have to have a certain degree of fortitude to keep on going in those types of situations. So I think I found licensing products to external companies doesn't work. Most often you get, you need somebody internal to a company who feels passionately about the product to make sure that it actually gets pushed all the way through. Really having people that feel that sort of passion and who are determined to make things work no matter what are really essential to driving these kinds of, certainly drug products through to the market.
Thank you.
Thank you very much, Dr. Cullis. It was very inspiring, and I love your contribution to the arts. I'll make sure to include that in my introduction next time. So and now for the concluding remarks, I'll invite Diana Royce to give these, and I'm pleased to introduce Diana Royce, who's the Executive Director of the Nanomedicines Innovation Network, or NMIN. She has over 30 years of experience in the post-secondary health and innovation sectors. She began her career in public service as a senior advisor on university policy and finance in Ontario, serving on provincial commissions and task force. Dr. Royce has served as the executive director of 3 networks of centers of excellence, or NCEs. First, HealNet, and then NCE based at McMaster University that focused on improving the use of data and evidence in health decision-making at all levels. And next, in Allergen, another NCE based at McMaster that focused on allergy genes and the environment, research and innovation. And finally, in NMIN, the NCE co-organizing this event with NanoCanada. NMIN is based at the University of British Columbia. And it's my pleasure to invite Dr. Royce to provide concluding remarks for today's event.
Thank you very much, Dr. Diorio. We now come to the close of what has been a fascinating, timely, and productive workshop focused on current and future strategies to build the Canadian nanomedicines ecosystem. Today's presentations clearly illustrate how, when faced with the global COVID-19 pandemic, Canadian scientists and startup companies, academia, and government labs rapidly pivoted their efforts to develop and commercialize novel diagnostics and therapeutic nanomedicine-based approaches that have global market potential. As noted, however, Canada still lacks a national sustainable strategy for developing and manufacturing vaccines and therapeutics in response to pandemics, and to leverage the world-class expertise in what today's opening keynote speaker, John Lewis, called the new era of genetic medicines. The Nanomedicines Innovation Network and NanoCanada's goal for this event was to bring together key stakeholders to explore current strengths, new market opportunities, critical nanomedicine sector ecosystem gaps to be filled, and ways to collectively overcome current barriers to scale up in order to extend Canadian companies' national and global market reach. Our three engaging keynote speakers helped us achieve this goal by highlighting strengths and successes to date, identifying promising future directions and opportunities, and underscored the need for increased support for scientific entrepreneurship and the identification of long-term and resilient strategies that support development of a Canadian nanomedicine manufacturing infrastructure and production capability. Nanomedicine approaches enable rapid, effective response capabilities to unmet medical needs and national and global public health emergencies. Facilitating expansion of Canada's nanomedicines ecosystems is a strategic initiative around which Canada's science and innovation community can unite. I hope every one of you leave today's event committed to participate in future meetings to establish a Canadian nanomedicines manufacturing cluster and move us from the visionary statements articulated today towards the development of a viable plan for collaborative cross-sectoral action. On behalf of NanoCanada and NMIN, I would like to thank our speakers, moderators, panelists, and attendees for making this an outstanding event. Please ensure that you sign up to receive NanoCanada and NMIN's emails so that we can keep you apprised of upcoming news and events of relevance to the nanomedicines community. And please also complete the post-event online survey. With that, I declare this workshop adjourned. Thanks for joining us, and let's keep this conversation going. Have a great afternoon.